Hello, good afternoon. We are going to just do a few questions from the Lecture 9 study guide, and we're going to finish up the lecture for number 9. And then we're going to do a little bit of number 10 today as well. So we're not going to do the full 2 hours because I know you do have that quiz to do for me. So I want you to have some time to prepare for that. You do have two chances to do the test today. So you might want to do it um, after class and then see what your mark is. And if you don't like the mark, study a little bit more and then try again later on in the evening. So this is the first question I want to do with you. It's again in the study guide for lecture 9. And it asks you some questions about the electrophoresis. Yeah, so here is a gel picture for the electrophoresis, which is a technique that we use to separate the DNA based on the length. And in this particular case, we have 4 lanes. Each of these letters represent a particular lane, and we load one sample into each lane. What we are loading is bacterial plasmid, so like circular DNA, that has been digested using restriction enzymes. Indicate the following on the gel, where the positive electrode and the negative electrode was located. So remember, these are the well. This is where the samples are going to be loaded at. And then we would have a negative terminal here and a positive terminal. And once you've connected them, the current is going to go from negative to positive. Okay, so this is the direction of current, which will help push the DNA through this network of fibers. And the smaller DNA is going to be able to weave through the network much easier compared to the longer ones. And so they would migrate further down the gel. So knowing that, we can find the location of the largest band, which would be the band that is closest to the well. So it's over here, right? That would be the longest DNA. We check all the lengths. There's nothing else that's going to be on top of it. So that would be our longest DNA. Whereas the shortest DNA, it appears to be over here in lane C. That's the one that is able to travel furthest down. Furthest down the lane. Okay. Next, the number of cuts that were made on the plasmid to produce this number of bands. If you take a look at a plasmid, and let's say we cut it 4 times, right? So cut it 3 times, and then maybe 4 times, and we actually gonna have 4 fragments. I'll draw it out for you. Right? We're gonna have this piece, this piece, this piece, and this piece, right? So that's 4 cuts in total, creating 1 band, 2 band, 3 bands, and 4 bands like that. So if you see 4 bands in lane A, that means there are 4 cuts. Okay, so this is for plasmid. If you cut a plasmid 4 times, you're gonna have minimum 4 fragments. Okay, and then you can cut it 3 times to get 3 bands, 5 bands here 5 times, and 4 times. Okay, so that's what happens when you are cutting the plasmid. Now it's a little bit different. The question says, will the answer be different if linear DNA was used instead? And it would be different. This is a linear DNA. If you cut it 3 times, right, cut it 3 times, then you already have 4 pieces, right? Okay. So if it's linear DNA, you need 1 cut less. Okay. I'll write down for you the number of cuts is 1 less then number of bands. So that's something that you have to pay attention to. All right. Any questions? All right, moving on. These questions are pretty standard. You can look at the study guide if you need to. I already post the answer for the study guide, so you can check that out yourself. I'm just going to be doing questions that requires a little bit of explanation. All right, next one here. We have two restriction enzyme, BAMH and BCL1, and we have a DNA fragment here. Okay, so the question is, how many DNA fragments would you expect if we cut the DNA using BAMH? Okay, so BAMH is right here. We are looking for the GGA-TTC. Okay, so just scan the sequence and see if you can find GGA-TTC. Hopefully you can find it, right? It's right here. GGA-TCC. So that means we're gonna have a cut right here. Okay. And then that's it. There's no more. GGA-TCC. That's all. If you treat the DNA fragments produced in part A with BCL1, okay, this is a different restriction enzyme now. BCL1 is TGA-TCA. So I'm gonna scan the sequence and look for TGA-TCA, which is right here. Right? So we're gonna have another cut. There you go. So now we're gonna load a different sample onto a gel, and we have to figure out what the gel is going to look like. So let me draw the gel here. We have 4 lanes. Lane number 1, lane number 2, 3, and 4. All right. So for the first lane, it says untreated, just DNA sample alone. So I'm gonna put like untreated. So if you just have the DNA and you don't cut it, then it's going to be the longest, right? Which means it's gonna be closest to the top, like this, right? This is uncut, is the original, is as long as it gets in this question, right? It's gonna be very close to the well. I hope that makes sense. Next, we are going to only use BAMH1. Okay, BAMH1. Now, you look at the way we cut this with BAMH, we're gonna have like a short segment here, right? Short DNA, and then we're gonna have a long one, right? Remember, we're only using BAMH, so the red cut is not gonna be there, right? So I'm gonna have a really, really short one at the bottom. Okay, I'm gonna color code it to help you understand this. Like this fragment actually correspond to the left fragment here, and then we're gonna have the other one. It's not gonna be as high as the uncut one, because you cut it, it's gonna get smaller. So that's why it's gonna be a little bit lower, right? So that's the BAMH alone. So we need to take a moment and think about what the next column is gonna look like. Okay, can you do that for me?
Excuse me, sir. I have a question.
Yes, please.
Um, For the BAM8, how do you know which segment is treated with the BAM8? Like there's two different parts. So how do
Okay, so there's only one part, right? Like this is the original sample. That's the original sample. And you're treating this sequence with BAMH, right? So if you're treating this with BAMH, you're only going to be cutting it over here, right? It's just one cut. That's all. So you're loading this into the second lane, which is why you're going to get a short band at the bottom. And then you're going to get like, you know, this longer one, which is closer to the top, right? But not as long as the uncut one. That's why it's going to be lower than the band from the first lane. Is that your
Sir,
question? Yeah.
to add to that question,
Mm-hmm.
is that given, you know, BAM-H, like where to cut it?
Oh yeah. On the
It's
test,
given.
if anytime I ask you to cut something, I will definitely give you the recognition site.
Okay,
Is
so
that
you're
what
gonna
you're
give
asking me?
us BAM-H, like this little chart that's on the left side here.
Yeah, like this one, right? It will always be in
Yeah,
the question. If you need it, it
question.
will be in the
Okay,
question. Yeah.
okay.
All right. Does that answer your question, BB?
Yes.
Okay. All right. So now we're just doing BCL1, right? BCL1. Okay. So BCL1, we're gonna cut here, right? Because it says TGA, TCA, right? So TGA, TCA is right here. So I cut right that. So now I have two pieces, right? I have, you know, like this yellow piece, and then I'm gonna have the rest of it, right? Just one cut because I'm only using BCL1, right? Now I'm just gonna put the other stuff back so you can see the yellow fragment here is actually longer than the green fragment. So when I draw it on the third lane, it should be a little bit above the green one. Right? This corresponds to the, you know, yellow section here. And then the other one is gonna be lower compared to this one. Right? It's lower because it's also shorter, right? Now, fortunately, you don't have to draw anything on the final test, but you do have to understand what I'm saying here to answer some of the questions, right? So if you do BAMH and BCL1 together, I am gonna get the yellow piece and I'm gonna get the green piece. Both will show up at the exact same location, right? But then I'm also gonna get the middle piece. Okay, that's gonna be somewhere here. Okay, because the middle piece is shorter than the other two fragments in the other lanes, right? So there you have it. That's how you would do it.
Do we have short answer questions on the final test?
Nope, just multiple choice and multi-select this time. All right.
When is it? The final test?
Final test is next Thursday. Next Thursday.
Okay.
Yeah. So we will finish up lecture 10 this Thursday. If we have time, we'll do a little bit of review as well. If not, I will post a review video for you. And then no class next Tuesday. You just study on your own. And then you will do the final exam, final test on the 14th. Okay. So that's the schedule. Are we okay?
Just a question about the BAM8 and the BCL. How, I don't understand how the first one is so it's at the position where it
Like this
is.
one? This one?
Yeah. Like
Okay.
I get the other two, but not that
Yeah,
one.
sure. So this piece is this middle guy, right? Do you agree?
Yes.
Okay. So let's compare to just the BCL1. Okay. If I just use BCL1, then it's the yellow and this whole piece that I'm putting a black thing around, right? A black circle, right?
Yes.
This black circle, this circle part correspond to this band, yes?
Yes.
Is the pink shorter than that?
No.
The pink
Oh,
part.
yes,
It
yes,
is, right?
yes.
That's why the pink is lower than this one, because it's shorter, right? If it's
Oh,
shorter, it's going to be able to go like further down the gel, right?
yes.
Does that answer
Okay.
your question now? Yeah, okay,
Yeah, okay.
good. All right. Anything else before I move on? All right. So that's pretty straightforward. You can do that. Okay, this is another one here that we should talk about. I already taught you this last time, but like super quick review. These are insertion sites, right? These are restriction enzymes that will cut the plasmid at those locations. If you treat your gene of interest, that's a gene you want to clone. That's a gene you want to like mass produce, right? Like a gene that makes insulin, for example. That if you want to put it in the plasmid, if you treat that gene with PST1 and you treat the plasmid with also PST1, PST1 is over here, right? Then you will create complementary sticky ends that will allow the gene to go into the plasmid. So you can insert using PST1 and the gene would go right here. You can insert using EcoR1 and the gene would go right here, and so on and so forth, right? So depending on which restriction enzyme you use on the plasmid and on the gene, you will be able to put them together, okay? Remember, you have to use the same restriction enzyme. The sticky end created by PST1 is not compatible with a sticky end created by EcoR1. Okay, they're gonna have non-complementary nucleotides. They won't stick together. So that's what the pink sites are for. All right, and then we have these one here, right? The AMPR and the TETR. Those are antibiotic resistant genes. It gives the bacteria antibiotic resistance if they accept the plasmid. So this gives us a way to determine whether the bacteria has been transformed or not. Okay, so that basically answers ABC. Okay, so let's take a look at this question here. Okay, this question. If you insert the gene by treating your DNA with BAMH and transform it into E. coli, that means like you feed it to them. What are the expected results if you grow the transformed E. coli in the following culture? Okay, if you grow them with no antibiotics, are they gonna grow or not grow? If you put them in growth culture with no antibiotics, just nutrients, grow or not grow? Yep, they will grow. Okay, if you don't give them antibiotics, they will grow. What if you culture them with tetracycline? Well, let's take a look. This is BAMH insertion. That means my gene is gonna go right here. That's the gene of interest, right? We are putting it right in the middle of tetracycline. You destroy the tetracycline resistant gene. Even if they have accepted the plasmid, they will not have tetracycline resistance. So if you grow them in the presence of tetracycline, you're gonna kill them. All right? What about ampicillin? Are they gonna grow in ampicillin? Yes or no? That's right, they will grow. No problem. The ampicillin gene is not disrupted. If they accepted the plasmid, they will grow in ampicillin. All right? All right, let's do another one here. This is the same question, but this time we are inserting using EcoR1. EcoR1. All right, do I have enough space here? Okay, doesn't really matter. Let's make it smaller. Here. EcoR1 means it's gonna go right here, right? Okay. So I want you to think about it and type me some answer. Okay, say yes, 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 no, 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 yes, no, yes, whatever it is. Okay, type your combination in the chat box, and then we will we'll talk about it. Okay. All right, we have a bunch of yeses. Okay, amazing. No antibiotic, yes, grow. Tetracycline, no problem. We got the tetracycline resistant gene. Okay. Ampicillin, no problem. We have the ampicillin resistant gene, right? As long as you're not like inserting something into the resistant gene, you're not going to be disrupting it. So you're going to be okay. Is that clear? Are you guys able to do this on the test? I hope so. Okay. I hope so. All right, let's do one more question here. Predict what will happen in each of the following situations during gel electrophoresis. Okay, so let me draw the gel again. And, you know, these are the wells. And what happens if you reverse the electron? So normally it's supposed to be positive to negative, normally it's supposed to be negative to positive. But if we reverse it, then the current's going to run backwards. All right, so what happens is the DNA will be moving in the opposite direction. Right? Sample, exit, samples, exit at the top. Lost. Okay. We'll lose the samples. Okay. Everything's gone. Just run backwards, right? It all escape at the top. Run the gel for too long. Diaphragm is no longer visible. Diaphragm is that like blue line that I showed you last class, right? That gives you an idea, an approximate location of where your DNA sample is going to be. But if you leave the current on for too long, so long that even the blue dye is like out of the gel, and what does it mean? Well, it means that samples are lost at the bottom. Okay, you've let the current run so long that even the short ones are going to be gone for sure. You might still have some long pieces. Okay, like remember the example we just did, the long piece was supposed to be here, but if you leave it on for so long, the long piece might be like all the way down here. Right? It's not that they can't go through the gel, right? I keep on telling you, it's just that it takes longer. So if you let it run for like too long, eventually, all the DNA will get through and they would just like exit from the gel at the bottom. Remember, the gel is like in this buffer solution, right? So when they come out from the other end, they just get lost in the solution. You can't really recover it. Okay. Forgot to add ethidium bromide. So DNA will be invisible under UV. And you can't even see them. Right? The ethidium bromide allows the DNA to fluoresce when you shine UV light at it, right? So if you forgot to add that, nothing will happen when you shine UV light at them. You can't see them. You can't detect them. That's all. All right, so I want to go through, like we still have a few slides left from the biotech stuff last time, and let's quickly go through it. Now we were doing gene therapy last time, and I just want to quickly remind you that we try to achieve a few things with gene therapy, right? First one is to replace a non-functional gene. We did this slide already, okay? I'm just giving you a quick review. So we can replace a non-functioning gene, right? If something is not working, you deliver a functional gene, and that should fix the problem, right? But these has to be recessive conditions. So when you introduce the normal gene, the normal gene is a dominant copy, which will override all the recessive ones in the cell. If it's a dominant disease, even if you introduce the functional gene will be covered, will be like silenced by the dominant effect of the disease gene, and it won't be useful. We can introduce a gene that would block the effects of an overly active gene, like an oncogene. Oncogene is like these, green lights that are always on, right? That tells the cell to keep on going through the cell cycle, keep on dividing and dividing, and we don't want that. So if you can introduce a blocking gene to suppress the oncogene, then you essentially shut off the green light and you slow the cell cycle, and the cancer cells will stop growing, theoretically. And then we can also insert a suicide gene into the cancer cell, cause them to make a toxic product that would kill them off. And alternatively, we can insert a marker gene that allows the cell to put all these signals on the cell membrane, and the signal will attract the white cells to come and destroy them. Okay, we can do it ex vivo, take the cells out of the body, modify them, and then put them back. Or we can do it in vivo, which is just deliver the DNA directly into the body. So what are some of the challenges associated with gene therapy? These are the challenges. Let's take a closer look at each one of them. Targeted delivery. Okay, we have a couple of problems. When we deliver the gene, we need the gene to go into the right cell, the correct kind of cell. Let's say you are trying to deliver a suicide gene. You need that to go into the cancer cell and not the normal cell. So how do you make sure, like when you inject it into the body, inject the DNA into the body, right? Like how do you control which cell takes it up and which cell don't, right? You only want the cancer cell to take it up, right? So that's a challenge, right? Obviously, if the suicide gene go into the wrong cell, not only will the therapy not work, but now you're killing healthy cells, and that could have its own problems, right? Even when the cell, when the DNA goes into the right cell, the gene needs to be turned on. Okay, and sometimes your cells don't like to have foreign stuff coming in, and they would shut them down. So, it's about finding the right target, and then going into the cell, turning it on, keeping it turned on. Okay, like not just on for a little bit and then it's gone, right? We need it to be on continuously, right? So when it comes to the target delivery aspect, people usually use virus because virus on their own, they have certain trophism, they have certain preference, okay? Not preference, but like they have certain target in the body, okay? For example, hepatitis virus, okay? What kind of cells does it target in the body? Do you know? You must have heard of hepatitis, right? What organ does it target?
Your liver?
That's right. It's not gonna affect your stomach, it's not gonna affect your throat. It will leave everything else alone. It will only have a preference for liver cell. Okay, so like if you want to deliver something to the liver, we can learn something about these, hepatitis virus, right? Cold virus, flu virus, they target the airway, right? Okay, the throat area. So, if you want to deliver something there, then you can use those virus, right? So of course they have to take out all the parts of the virus that actually causes the disease, and you just leave the shell and replace it with the gene that you like. Okay, if all goes well, the virus will go and deliver the gene right into the cell that you wanted to deliver to. But since we're using viruses, there are certain immune response that could occur. And in researching gene therapy, people try to minimize the immune response. We don't want the immune system to, overly react to these things. And sometimes the immune system uh, would react to the cell after it has accepted the gene, because you have changed it in an unexpected way, right? So there's a lot of moving pieces here that needs to be considered. Interaction with other genes. Okay, so let's say we found the right cell, we are able to deliver the gene, but what if this gene insert itself in the middle of another gene, right? Like we did with the plasmid, right? So let's say there is a tumor suppressor gene. In case you forgot, tumor suppressor genes are red lights, right? They stop the progression of cell division under the right circumstances. And if we use gene therapy and it just insert itself right into the middle of a tumor suppressor gene, then you have broken the red light essentially, right? Then now there's no red light. The cell is gonna divide, potentially in an unchecked manner, and that could be problematic. Okay. Cost. Many of these genetic disorders are quite rare, and so there is not enough funding to fund them. Things like sickle cell anemia, beta thalassemia, hemophilia, those are really common. So it's not surprising that there is a lot of successful cases of gene therapy for those situations, because there's a lot of funding for it, right? If they invent something that is suitable for everybody, then the market for curing sickle cell anemia is huge. There's a lot of money in it, right? For pharmaceutical companies to invest in these things. But then for rare genetic conditions that are more rare, like only affects a small number of individuals, there's no incentive to invest a lot of money into it, right? So like if they create some kind of therapy, it would be a tailored approach for people. Now, there is this guy. He was on Americans Got Talent, or maybe Britain Got Talent, or some one of those shows. Okay, those like performance show. But he was born with a genetic condition that caused him to be blind. All right. But he's very talented in the singing. I think he ultimately won the contest. And what happened was, after all the fame that he got, he was able to crowdfund and get enough money to do gene therapy to fix his blindness, and it actually worked. But the cost is very, very high. The eye treatment costs $850,000 for both eyes, which is just a little bit under $500,000 per eye, and that's seemed to be the general case for gene therapy, ranging from $500,000 all the way to well over $1 million. And depending on the kind of gene therapy we're looking at, some gene therapy requires ongoing treatment. So something called like spinal muscular atrophy, which is like shrinking of muscles associated with the spine, that causes $750,000 in the first year, and each year you will have to shell out more money. And that is for life. And these are like US dollars, right? Not even Canadian dollars, right? So like it's like even more expensive if you put that into that context. So not everybody can afford it, right? Some drug companies say, we have to pay only if it works approach, right? So if it doesn't work, that's okay. You can keep the money, right? But then what considered as works can be tricky. Like what if it works for the first year and then it stopped working? Right? Do I get my money back, or like what's the situation there, right? So it's not as simple as that. And a lot of insurance company, of course, would not be covering things like these, right? Even if they would, they wouldn't be covering 100% of it. It's just like it doesn't make sense for their business model to do that, right? So a lot of people turn to crowdfunding as a way to raise enough money to get on these gene therapies, and a lot of them are still experimental, right? At best, it of course can fix the problem, but a lot of times they might not end up working, and sometimes in unforeseen situations, they might cause problems to the individual's health as well. So that is the biotechnology unit. Okay, you do have a quiz to do today, which is 8 and 9. I saw that nobody has done it yet, which is surprising. Usually a few people would do it like bright and early, like 6 a.m. in the morning. But that's okay. I mean, you guys probably need some time to study for it. Now, any other questions before I do a little bit of number 10 with you? Everybody okay here?
Sir, all this stuff that you just
Yeah, did yeah.
is it on the quiz?
Like the theory stuff, like with the challenges with the gene therapy and stuff like that. Is that what you're
Yeah.
asking? Yeah, maybe just 1 or 2. If you have been listening to me, you would get it. Okay, like I was saying, which of the following is a challenge for gene therapy? And then you would choose like cost, affordability, target delivery. Just
Okay.
yeah, basic stuff. Nothing too obscure. Okay, but like these stuff, right? Which we learned before,
Mhm.
you those are definitely on, right?
Okay.
Okay. Don't worry, you have two attempts. Okay, 2 attempts. Take advantage of that. All right, I won't keep you too long. I just gonna go through a few slides here. Body tissues. All right, so next semester you will be taking the anatomy and physiology, which is a direct continuation from this course. That one you would have 6 hours per week. Are you guys planning to stay online, or are any of you going back to in person? Like, what's the deal here? Do you guys know?
What do you recommend that we do?
I recommend you do whatever makes you feel good. Okay, like if you have been finding the online experience to be suitable for your lifestyle, right? Then continue, right? I mean, online has a flexibility, right? You don't have to come necessarily to the synchronous session. It's good if you have jobs, if you have family to take care of, right? So, yeah, but the drawback is like you don't have a community necessarily, right? Like you don't see other people. And sometimes it's hard. Like for me, like if I have to do an online course, personally, I find it difficult to do because I sometimes don't have the self-discipline, right, to keep up with the work, and then it just keeps on piling up, right? Are you able to do a mix though, Patricia? I think for the pre-health, you either do one or the other, right? Other than the English, that one we have no control over. But then, yeah,
English is in class, so right after this, I get straight to the campus.
right. Oh, in the afternoon, they still have another class in the afternoon.
Yeah, I do English in the evening.
Oh, I see.
You
Yeah.
have to do English in campus?
You don't have to, but I choose to because I'm just alone at home, just to communicate and see other people.
Oh yeah, okay.
So that's why I chose it like that.
Yeah, so there is definitely pros and cons for both, right? And I mean, it's the winter is coming. Some people don't like to commute, right? But anyway, if you want to know, I am doing one of the online sections for next semester for the anatomy physiology. If you want to be in my class, I think my code is 201. If you don't want to be in my class, then don't choose 201. Okay, either way, it doesn't matter. I'm just telling you, you make your own decision. All right.
I just want to ask something.
Yeah, Blessing, go ahead.
Do we have option to choose if we want to be online or in person?
I would assume so, right? Like when you choose your course, you can do that, right? Can't you?
Oh, I don't
Yeah.
know.
Yeah, but then for some subjects, like for English, I think it's depends what they offer, right? I'm not 100% sure about what happens in English, but I think for the pre-health, there are options available.
Your
Anyway,
code is 201.
what's that? Sorry?
You said your course is 201.
Yeah, yeah, unless they change it, okay. But as far as I know, for now, it's yeah, it's 201. Okay, that's the section number. I mean, you guys are really, I really enjoy having, you know, the 10 of you. Every time you guys show up, it's the same 10 people. So I think you guys are very dedicated. I mean, I'm sure people also watch the recordings, but you know, coming to the live section, it's a little bit different, right? Anyway, so the body tissue, this is a connection with the next semester stuff. So let's start with stem cells. Okay, stem cells are cells that can undergo mitosis to make more stem cells. Okay, so like you're not going to run out of stem cells. But in addition to making copies of themselves, stem cells can also differentiate into specialized cell type. You will hear this word quite a bit moving forward. To differentiate is to commit to a particular cell type. Okay, it changes, transform into a particular cell type. Could be liver cell, could be brain cells, could be stomach cells. Okay, whatever it differentiate into, that final cell would have a specialized function. And it's a one-way transformation. There is no coming back. Okay, so when the baby is still in the womb, right, we have these embryonic stem cells. These are very versatile. They can become virtually any cells of the body under the right condition. And they will, right? Some will become brain cells, some will become cells in the eyes, some will become, you know, cells in the kidneys and so on and so forth. That's how we get the different cell types. They all came from embryonic stem cells. However, after birth, the number of stem cells are going to be reduced, and they tend to be restricted to certain areas of the body. Like even in adults, we have stem cells. We have stem cells in the bone marrow, but those are only good to become blood cells. Okay, they're not, you know, as versatile as the embryonic stem cells. You can't take a stem cell from the bone marrow and turn it into a brain cell. It doesn't work like that anymore. They're more committed by that time. Okay, it's like us, right? We are in the pre-health program. We're committed to go on to healthcare related fields, right? Practical nursing, right? Pharmacy technician, whatever the case is, right? We're not going to go into, you know, accounting, right? That's like another program, right? So similarly, stem cells that you find in the bone marrow will be restricted to becoming cells in the blood. Whereas if you find stem cells in the intestinal tract, they would be committed to different cells of the GI tract, right? So it's less versatile after birth. So that's why people sometimes go for the option of keeping the embryonic stem cells from their baby when they're born, right? There's a little bit that you can collect from the umbilical cord. It's called the cord blood. And you know, if you can afford it, you can pay to store that in a -80 freezer, and one day, you know, if your child ever needs that for whatever reason, it's going to be available. Okay, but it's expensive, right? Not everybody can afford it, and you know, not everybody necessarily wants to do that either, right? We were offered the choice to do it. I didn't do it for my kids. It was like ridiculously expensive. Okay, so this picture shows you the stem cells can, you know, make more stem cells, and then they can differentiate, right, to become different cell type. Okay, brain cells, liver cells, whatever the situation is. So throughout this course, we actually started at the atomic level at the beginning, right? That seems like so long ago, right? It's the beginning of September. We learn about the atom that makes up the body, right? Carbon, hydrogen, oxygen, nitrogen, phosphorus, right? And all this thing can come together to make molecules. And then macromolecules we learned, and then we also learned about the different organelles, and then we learned about all the cellular processes that are happening, DNA replication, transcription, translation, right? All those things. So now it's time to move up yet another step in the hierarchy. Okay, so tissue, they are basically different cell types that have assembled into a group, and it's going to be functioning together to achieve a specific function. Okay, so for example, we have cells that will be able to do storage of energy. We have cells that are able to create motion. We have cells that are specialized in connecting things. So all these things could come together, and we would have a tissue. Okay. Example would be like fat tissue, muscle tissue. There are 4 types of tissues that we'll be learning in the lesson. And then these tissues can come together to form an organ, and then the organ can come together to form organ system. So these last 2 points is basically the whole course next semester. Okay, other than the skin, which is this integumentary system, other than that, we're going to be learning all of the other ones. Okay, basically going to learn 1 system per week. Okay, 1 system per week, starting with the cardiovascular system. Okay. Now, our body has many cavities in it, actually, many empty spaces. You know, the organs are found, different organs are found in these different locations. I'm just going to show you a picture of it. It's probably easier that way. So up in the head here, we have the cranial cavity. You don't need me to tell you. I'm sure you know that the brain is in there. Okay, that's where the cranial cavity is, and that's where you hold your brain. We have a vertebral canal that goes all the way down to your lower back, and in there, you have your spinal cord. The spinal cord is really small. Most people don't know it. Spinal cord is no thicker than your finger. Okay, it's really, really tiny, very delicate. That's why it's protected by your backbone, right? So the thoracic cavity is right here, the chest. Okay, so tell me, give me 2 organs that are found inside the thoracic cavity. You must know, right?
Your heart.
Yep, heart is one of them, slightly to the left. What else?
Stomach.
I'm sorry, what's that?
Your stomach.
Stomach is a little bit lower. We'll get to that later. What's another thing that will be fine? Inside the chest. That's right, Justine, the lungs. You have 2 lungs. Right? Left one and the right one. The right one has 3 slices, and the left one only has 2 slices. So these are in the thoracic cavity. And then there's the diaphragm. The diaphragm is like a sheet of muscle right here that separates the thoracic cavity from the abdominal cavity. Okay, the diaphragm helps you breathe. It moves up and down as you're breathing. And when you have hiccups, that's when your diaphragm is like going crazy. It's like poop, like that. Like it's doing these kind of like spasm. That gives you the hiccups, right? So in the abdominal cavity, okay, lots of stuff in the abdominal cavity. Give me like 4 things. Okay, tell me something, anything that you think it's in the guts, right, as they say. What have we?
Pancreas.
Excellent. Pancreas. That's the one that will be making the insulin, right? We talk about that in the biotech stuff. Anything else? Pancreas is on the left side. Yeah.
The liver.
The liver is on the right side. Very good. Anything else?
Spleen.
The spleen. Oh, that's a rare one. People usually don't talk about the spleen. Yes, you got a tiny thing called the spleen, which is tucked right behind your stomach. It's your immune organ. It also helps you store blood. Okay. That's right, the intestine. Bunch of things here. Very good. Okay. So like some test question might be asking you, which of the following is in the abdominal cavity. Nothing weird, okay? Like I'm just gonna give you like well-known stuff. Okay, I'm not gonna put like the something that you never heard of before. Okay, it's like obvious things. Okay, further down we have the pelvic cavity. Okay, this one's a little bit harder. Okay, so like let's go with 3 things. Okay, give me one thing that's in both male and female, and then give me one thing that's only found in female, and then the other only found in male.
Ovaries.
Ovaries, that's right. That's only in females. Yep. And then what's another one? Bladder. Okay, that's good. But we want to say urinary bladder because there is an other bladder in the body. Okay, the gallbladder, right? Like fish, they have like swimming bladder, right? So like we want to be specific. Urinary bladder. Okay, so I want one thing that's only found in guys. Only found in male. Testicle is like outside of the body. Like this is the penis, and then the testicle is like down here, right? So we want something to be like in the pelvic cavity. In the pelvic cavity. If you are age 40 and above, your doctor might be checking that for you.
Prostate.
That's right. Very good. Okay, prostate. All right, so those are some of the things that will be found in the pelvic cavity. Okay. Excellent. So next we're going to learn about the anatomical terms. Okay, anatomical terms. These are terms that we use to describe the location of body parts relative to each other, and for that you need to have your study guide. Okay, so I'm looking for this picture right here in the study guide, and while you retrieve that, I will give you a little bit of an explanation here. So the way that the person is standing there is called the anatomical position. Right? So we want to use these terms so that when we say something is on top of something else, we want to make sure that we have the right image in our head, right? Like the person is not lying down, or like he's not on his side, or like she's not upside down or anything like that. Like the image that comes to mind when you describe body parts is this picture. Okay, so this standing facing forward, hands a little bit apart. This is called the anatomical position. Okay. And for this diagram, we're just looking at it from the side, it's all. Okay. So these terms usually comes in pairs, describing the opposite thing. So if it's higher up, we say superior. Superior. Opposite of superior is what? You can guess.
Inferior.
Yeah, that's it. Inferior. Okay. So same thing here. Superior, inferior. So let's take a look at some examples. Okay. Think of the picture, okay, that you just saw. In that position, in the anatomical position, the hands are blank to the feet. What do you think, superior or inferior?
Superior.
Yeah, superior. It's higher up. The knees are blank to the waist.
Inferior.
Below, right? Elbow is blank to the wrist.
Superior.
Superior, that's right. Right. That's why like we have to think about it in that picture, right? It's not like with your hand raised up, Because if with your hand raised up, then elbow is below the wrist, right? Like you have to think about it in that picture. The calf muscle is blank to the ankle. Superior, that's right. Okay, for those of you who don't know where the calf is, like if this is your feet, okay, the calf is right here. Calf muscle. Okay, ankle is down here. All right. Next, anterior and posterior. So over here, in front, that's anterior. And the back, that's posterior. Okay. And we have another alternative way of describing this. If it's on the side of the tummy, okay, we say ventral. If it's at the back, then we say dorsal. Okay, so you might wonder, hey, why do we complicate things? Why can't you just use the same thing? Why do we have to have two sets of terms? Well, it turns out, like if you think about like an animal, right? Okay. Like a dog or something. Okay. So for our four-legged friend, the anterior is this way, and this is posterior, just as you would expect, right? But where is the tummy? Where is the ventral side? Huh? Where is it? It's pointing straight down, isn't it? And then that would be pointing straight up, would be the dorsal. Okay, so it has to do with the the natural orientation of the organism, right? When we use some of these terms. So if you think of like think of the last shark movie that you watch. Okay. Like when you see this thing coming out of the water, you know it's time to run, right? Or to swim away. What do you think this fin is called on the shark?
The dorsal fin.
That's right, the dorsal fin is exactly what it's called. It's on the back. Okay. That's the only part they ever show. It's the cheapest thing to make, right? Rather than the whole shark. Before CGI was possible, they only make the triangle, okay? The dorsal fin. So when we describe the nervous system later on, right, we use these terms, dorsal and ventral. That's primarily where it's that's primarily where you're going to see it. All right, so let's see. Complete these sentences with anterior or posterior. Okay. So I give you a moment to try them out. All right, the heel to the toes. What do you think? Anterior or posterior?
Posterior.
Posterior is correct. Okay. Bicep is here. Tricep is here.
Anterior.
Okay. Anterior is correct. Do you guys know your hamstring and quadriceps? Okay, I gotta stand up for this. Okay, like this. This here, that's the quadriceps. The back, that's the hamstring, okay? So the hamstring is blank to the quadriceps. What is it?
Posterior.
Posterior. Okay, people say the quads, right, and the hammy. That's what they are. Quads is in the front, hamstrings in the back. Okay, next. I hope this is more refreshing for you, right? Like after learning all the like biotech stuff, this should be more fun, right? I hope. Fun is a relative term. If we divide a body right down the middle with an imaginary line, that is called the midline. Midline. It's essentially a line in the middle. Midline. So things that are further to the sides of the midline, we say lateral. And things that are like closer to the midline, closer to the middle, we say medial. The eyes are medial to the ears. Okay. Eyes are medial to the ears. And then the eyes are lateral to the nose. Right? Do you understand that? If it's lateral, it's further out. If it's medial, it's further in. It's closer in. Okay. So here we go. The arms are lateral to the midline. The neck is on the midline, right? Okay, like there are a couple of things that are on the midline. The neck, the mouth, the nose, right? Belly button. Okay, all those things are in the middle. So the neck is medial to the arms. Okay, neck is as medial as it gets. There's nothing more centered. The shoulders are blank to the midline, lateral. The ears are lateral to the nose. Are we okay with the lateral medial? All right. Next, we talk about structures that are either on the hand, on the arm, the whole arm, right, or the leg. And we compare it to the point where the limb connects to the body. So for the arm is connected to the shoulder, at the shoulder, right? The arm is connected to the body at the shoulder. The leg is connected to the body at the hip. So if something on the arm is closer to the shoulder, then we say proximal. If it's further away, then we say distal. Okay. Same thing on the leg. Proximal, distal.
Excuse me, sir.
Yeah.
Can we refer to those body parts that are in between as media also?
Say that one more time.
Like the body parts. I don't know how to say, I don't know how to explain.
Okay,
Can
I
we
think
call
this is what
those-
you're
those-
asking.
Yes, okay.
It's actually possible to have more than one correct answers. Okay.
Okay.
So for example, let's say the nose and the eyes, okay? The nose and the eyes. Okay. I can say the eyes are lateral to the nose. You agree?
Yeah. Yes.
Yeah. Can someone give me another correct term? If I describe the eyes with the nose, what's another correct term to use here? Besides lateral.
Superior.
Yeah, the eyes are superior to the nose, right? So like it's not like you have to use a particular word for a particular structure. There are multiple correct answers, but some answers are more appropriate than others. Okay, so for example, when we talk about proximal distal, right? Okay, we can say that the elbow is proximal to the wrist. Right? Even though it's correct to say the elbow is superior to the wrist, but because we're talking about things that are on the arm, it makes more sense to use proximal and distal. Is that close to what you were asking, or like I'm totally off here?
Yes.
Okay, yeah. Bibi, you had a question. I saw you on mute.
Yeah. I was just thinking, like, so for example, a certain body part, like the chest area, you don't have to say that, okay, the chest is superior to the knees or something. You can also say like it's lateral to the arms. Like it's not one
Well, the
term.
chest is medial to the arms, though, right?
Oh.
Because it's closer to the middle than the arms, right?
Yes,
Right.
yes,
Yeah.
yes. So you can use different words to
Exactly.
describe it. That's what I meant.
That's
Okay.
right. That's right. Yeah. So like the whole point of this is, you know, next semester when I say, you know, the trachea is anterior to the esophagus, then you know what I mean is, oh, the trachea is in front of the esophagus. That's basically what it is, right? Like, so like you will see these terms being used in different contexts, and you know, you kind of have to understand, right? Like I would say, oh, the distal end of the trachea is connected to the bronchus. Then you have to understand, oh, distal means like further away, right? So yeah, I mean, don't like get too hung up on it at this moment. I guess like you will understand when they're being used in the right context. For now, just worry about these examples. If you're able to do these fill in the blanks, you're okay. All right, fingers and to elbows. What is it? Are the fingers further down the arm or closer to the shoulder? What's going on here? Distal or proximal?
Distal?
Proximal.
Yeah, fingers are distal. Okay, look. If you look at the arm, right? This is the shoulder. Fingers are further away from the shoulder than the elbows are. Yeah, if it's further down the limb, then it's distal. Okay. If it's closer to the joint that connects to the body, when I say joint, I mean the shoulder, right? I know like elbow is a joint as well, but we are talking about like the joint that connects the limb to the trunk, right? To the body. So fingers are further away. That's why it's distal. How about knees and feet? Proximal, that's right. It's closer to the hip, right? The knees are closer to the hip than the feet are, right? What about wrists?
Proximal.
And fingers, yeah, proximal. That's it. Okay, if you don't understand or any further explanation, just ask. I have 2 more terms to go through with you. Everybody happy? Okay. Next 2 terms. Ipsilateral, that's how you say it. Ipsilateral or contralateral. Ipsi means same side. Contra is opposite side of the body. If both things are on the left side, then they are ipsilateral. Okay, like my right ear is contralateral to my left shoulder. Okay, although that's a weird way of using the word, but that's what it is. Okay. So the spleen, someone mentioned the spleen earlier. I think it was Patrice. The spleen and the gallbladder. Okay, let's take a look at the picture. The spleen is right here. It's a tiny thing here. and a gallbladder. Turns out people can live without a spleen and without a gallbladder. Fancy that. But what's the relationship between the spleen and the gallbladder? Are they ipsi or are they contralateral?
Contralateral.
They are contra, that's right. Very nice. What about the stomach and the liver? Stomach and the liver.
Contra.
Also contra, right? Stomach is over here, liver is over here. Opposite side. Contralateral.
Well, we have right lobe of liver and left lobe of liver.
Oh, I see what you mean. Yeah, but even though we have left lobe and right lobe of the liver, like the whole liver is primarily on the right side, right? Even though there's a left lobe, but like the whole thing is like, I see what you mean though. Yeah. The bulk of it, the majority of the organ. The liver and the gallbladder would be ipsilateral though, right? Okay. Anybody know what this little thing is? This you can also live without. In fact, nobody
The
knows what it
appendix.
does. That's right, that's the appendix. Right? So appendix is contralateral with the spleen, right? But ipsilateral with the gallbladder there. Okay. All right. Last pair, deep and superficial. Deep is exactly what it sounds like. If something is deep in the body, it's like buried in the core. If something is superficial, then it's closer to the surface. So bones are generally blank to the muscle.
Superficial.
Are bones usually outside of muscle, or are they usually inside muscle? When you think about eating like a chicken wing or something, right? Like it's the meat outside of the bone or the bone outside of the meat.
Meat.
Yeah, meat
Outside.
is outside. So that means the bone is like deeper, right?
People.
Does that make sense?
Yes.
All right, what about skin? Skin is blank to the muscle. Anyone?
Superficial.
Yeah, superficial. Nothing is more superficial than the skin, right? Other than perhaps like hair, okay? But like, its skin is pretty superficial when it comes to, layers in the body. Cranium, right? Cranium is right here, right? It's part of the skull. So the brain is blank. The cranium. Yeah, it's deeper than. Right. At least I hope so. The cranium protects the brain. All right, last one, the pericardium. And you're like, what is that? I never heard of it. Don't worry, just keep reading. The pericardium is a membrane that surrounds the heart. So we can say the pericardium is superficial, right? Because it wraps around the heart, right? Surrounds the heart. It's outside of the heart. It's superficial. Okay. That's all. That's all. Okay, I'm just gonna go through a few other concepts with you. We'll be done in about 5 minutes. So in next semester, I'm gonna be showing you a lot of pictures and diagrams we're gonna be labeling. Let me show you maybe some like brain stuff here. Maybe lecture 6 or something. Okay, so like this is a brain, right? So like you have to know what view is that? Like is that from the side? Is that from the top? Is that from the bottom? Like I would have other brains laid like this, right? You have to know this is like a superior view. We're looking at it from the top. right? And like, is it cut open? Is it not cut open, right? So that's why we have to learn these terms. And when it comes to cutting the body, you can cut it with different planes. Okay? So for example, we can okay, we can take a plane and then we can cut this way. Okay, so like using a plane that's parallel to the wall behind me, you can cut the body, right, to separate into front and back. So that plane that does the cutting is this one right here. Okay, so that is called the frontal or coronal. Right, so that divides the body into anterior and posterior. Okay, you might have heard of like a CT scan, right? Or like an MRI, CAT scan. All these things scan the body as slices. All right. That's frontal. You can cut it this way. Right? You can cut it like this way, like this. Okay. Cut it parallel to the ceiling or the floor. That is going to separate the plane into, separate the body rather, into superior and inferior portion, top and bottom, right? So there it is. That is a transverse plane. Plane. A plane is just like think of like a piece of cardboard, okay? That's a plane. We can also cut this way. Like this. Okay. Cut. That's like parallel to the wall on the side. And it's gonna be this one. There we go. That is the sagittal plane. Okay. Now I'm gonna put in a bracket here. You can say mid sagittal if you cut right down the center. That's a mid sagittal. But it doesn't have to be right down the center, right? It could be like off to the side as well, right? If you've ever been to the Science Center, there is a section for like human bodies, and they actually have a display where it's the body that has been cut, right, and fixed into like little glass plates of the different sections of the body. So like a transverse section of like, you know, what it looks like from the abdomen, right? So that is something that, you know, it's relevant to what you will be learning. Okay, so that is it for now. That's just a description of the planes. And then this is just to show you, you know, what it would look like if you actually cut with those planes. All right. And that's where we have 2 more questions. Let's do these questions and then we're gonna go. Select all the correct answers for the blank. The right thumb is something to the left big toe. Okay, so take a look at all these terms and see which one applies.
Lateral and anterior.
Lateral, okay, that's a good one. And what?
Anterior.
And anterior is the thumb in front of the toes though?
Yeah.
In the anatomical position? I don't think so, right? Like if anything it's like the thumb is behind the toes, right? If you're standing in the anatomical position. Like, or they might be at the same location, right? I don't think anterior is the correct one. Anyone else? Besides
It's
lateral?
superior.
It's superior, yeah, that's definitely it. And one more, maybe.
Contralateral.
That's right. One is on the right, the other is on the left. Okay, so like I said, there are more than one correct answer in a lot of these situations. Okay. All right, this one, we had a little bit of fun with this in the other class. Right? If you buy a piece of salmon steak from the supermarket, what cut is it? What cut is the fish? What do you say?
Is it Corona?
Yeah.
Corono? Yeah, that's
Front
right.
one.
Okay.
Transfer.
Corono. Okay. If you cut the fish this way, right, into front and
Mhm.
back, yeah, then you get yourself a nice piece of salmon steak, right? Okay. When you are filleting the salmon, that would be sagittal. Okay. Think about it. Right? Filleting is sagittal. I have not seen someone transversely cut a fish before. To cut it transversely would be like, I don't know how to, like you would hold the fish in the regular swimming position, and then you would cut it this way, right? Yeah, I don't think I've seen a cut like that before. Okay. All right, we good? That's all for today. Okay. Good luck with the test. And remember, I'll see you again on Thursday to finish up lecture 10. We probably will start a little bit of review for the final test. We won't meet again next Tuesday. You can of course find me if you need me to help you with the test. Okay. And then the final test is just gonna be on the 14th. Okay. Thank you for coming.
Sir,
Thank you, sir.
1 second. You said
Sure,
we're
sure.
not seeing you on Tuesday?
No, you will not be. We don't have a class on